Today I'm in the office of Dr. Carol Milligan, who's the director of the ALS Translational Science Unit. Many of you understand the importance of fundamental basic research. Many of our investigators are really focused on understanding how normal cells work, how they become abnormal in certain disease states. Other investigators are taking their basic science discoveries and trying to translate them to the improvement of human disease. ALS is a very important disease, and Dr. Milligan's research team is very focused on applying their basic research models to finding a cure for this disease. Carol, tell us a little bit about what happens in your research unit.
Okay, so our research unit, I want to start by saying it's more than one laboratory. It incorporates 3 or 4 laboratories, actually, and that includes Dr. Ron Oppenheim in our department, Osvaldo Del Bono in internal medicine, and Tom Smith up in orthopedic surgery. All have contributed in important aspects of the project. We are specifically interested in understanding what happens in ALS. We know that the motor neurons, the cells that sit in your spinal cord and innervate your musculature, become dysfunctional and eventually die. Using our mouse model, we've realized that pathological changes in this animal occur very early, much sooner, almost 3 months sooner than symptoms become obvious. That points out to us how important it is to understand what's going on in the disease, what's going on early, so that we can develop therapeutics that will interfere with those processes and therefore either delay or hopefully prevent progression of the disease. It's also important with our patients because right now there is no clear diagnostic for ALS. When everything else is ruled out, then they can rule in ALS. So many labs, including our lab, are trying to find biomarkers of the disease using patient material. One of our projects that highlights bench to bedside progression is a project that we've done looking at the role of heat shock protein 70 in preventing progression of ALS in the mouse model. Heat shock proteins are proteins that are normally found within your cells, and when those cells are subjected to a stress, they have increased expression. So what did we do with this? We took the protein and we added it to cells in culture and found that it promoted their survival in culture, and that was using embryonic chick motor neurons. It promoted survival during normal development of motor neurons, and then we put it into the mouse model. And in the mouse model, it delayed symptom onset and it promoted survival. We also found that following treatment with this protein, what it did was keep the motor neuron in contact with the muscle longer. So the animals had better behavior, better muscle strength throughout the progression of the disease.
So this is really a team sport, isn't it, Carol?
Yes, it is.
Thanks for all the great work you and your team are doing.
Okay, thank you.